Hello friends, in this video lecture we will be talking about the life cycle of T4 bacteriophage which exhibits the lytic cycle.
Let us now move on to the life cycle of the T4 bacteriophage, that is the lytic cycle. We have already seen the structure of T4 bacteriophage. It is a phage whose host is E. coli. T4 bacteriophage starts its life cycle with attachment to the surface of the host cell, followed by injection of the viral nucleic acid, replication using the host cell machinery, protein synthesis, assembly of the new phage particles, and finally, lysis of the host cell. Since it results in the lysis of the host cell, it is also called the virulent cycle. The first step in infection is the adsorption of the phage to the host cell. This is as a result of random collisions between the host cells and phage particles. The attachment is specific and is through specific surface structures called receptor sites. The specific receptors can be lipopolysaccharides and proteins on the cell wall, teichoic acids, flagella, or villi. The adsorption initially is reversible when only the tips of the tail fibers attach to the cell surface and is termed landing, as can be seen in the picture. If the physicochemical structures in the tip of the tail fibers correspond to the specific receptor sites in the cell wall, the tail is adsorbed. Thus, tail fibers play an important role in adsorption. The reversible attachment is followed by the settling of the base plate on the surface. This is due to the attachment of the 6 tail pins on the cell surface and is therefore called pinning. This stage symbolizes the irreversible stage of adsorption. Once the phage is irreversibly attached to the cell surface, it is now committed to the infection. The next stage is penetration. This involves 2 stages. The first is tail contraction. Once the attachment becomes irreversible, a conformational change occurs in the tail sheath. The tail sheath, which was made up of 24 rings of 6 subunits each, rearranges to form 12 rings of 12 subunits each. As a result, the sheath becomes shorter and wider, and the protein tube is pushed through the bacterial cell wall, as can be seen in this picture. Thus, it behaves like a hypodermic needle. The penetration is mainly mechanical, but it may also be aided by the protein G5 present on the base plate. This gene protein 5 has lysozyme activity, which makes an opening through the cell wall and also through the cell membrane. The second stage in penetration is nucleic acid injection. The nucleic acid of the phage is then forcefully injected through this opening. It passes from the head through the hollow tail and enters the bacterial cell, as can be seen in the picture. The remainder of the phage remains on the outside of the bacterium and is called ghost phage. Once the phage DNA enters the host cell, its transcription starts and takes place in several stages, leading to the formation of immediate early mRNAs, delayed early mRNAs, and late mRNAs. Immediate early genes are transcribed immediately using the existing host cell RNA polymerase. These genes code for the enzymes that break the host DNA. Delayed early genes code for enzymes like polymerases and ligases needed for replication and also an RNA polymerase that will transcribe the late genes. The late genes encode for the structural proteins of the phage particles and lysozyme required for the lysis later on. The next stage is destruction of the host DNA. Once the phage DNA is injected into the bacterial cell, the bacterial replication, transcription and translation comes to an halt and the host DNA is degraded. This results in the complete arrest of the host cell mechanism. The host protein synthesis stops 3 minutes after infection. The host DNA synthesis continues for up to 10 minutes, but it is mainly repair synthesis in response to the damage caused by the phage. The host cell arrest is mainly due to DNA fragmentation. It's time now for the next stage of phage DNA replication. The phage DNA replication starts about 6 minutes after infection. The replication cycle follows the usual steps of priming, elongation and ligation. Replication is bidirectional from a fixed origin. Leading strand is synthesized continuously and lagging strand is synthesized discontinuously in the form of Okazaki fragments. These fragments are then ligated by a DNA ligase. Sometimes the phage DNA is replicated using the rolling circle mechanism. This leads to the formation of long concatemers. Unit lengths of the phage DNA are then cut and are circularized. The next stage is maturation. Late proteins are synthesized around 9 minutes after infection. The phage progeny DNA duplexes and structural proteins now get accumulated in the bacterial cell. They start getting assembled to form the phage particles. The process of assembly of the phage particles from their components is called maturation. The assembly of the phage particles does not take place randomly, but in a well-defined manner. The head, tail fibers and base plate get assembled in 4 different sub-assembly lines completely and then combined to form the complete phage particle as can be seen in the picture on the right. The first step in maturation is head assembly. The assembly of the head takes place in 4 stages: prohead 1, prohead 2, prohead 3 and mature head. Prohead 1 and prohead 2 are the stages which forms the basic core and do not contain any DNA. During prohead 3 formation, the DNA starts getting packaged in the prohead. The mature head is completely filled with DNA. After the head assembly comes the tail assembly. The tail is assembled separately with the assembly of base plate, tail and tail fibers. The base plate on formation is joined by the core tube and core sheath. Meanwhile, in another subassembly line, the head is formed and it spontaneously joins to the tail via the head-tail connector, the collar. The tail fibers need not be present for this union. The tail fibers are separately formed in another subassembly line. Each tail fiber is made up of 2 parts, the proximal half fiber and the distal half fiber. They are then attached to the base plate. Thus, the first complete T4 bacteriophage particle appears after about 15 to 20 minutes of infection. The last stage is release or lysis. At the end of the intracellular phase, the host cell lysis takes place. In the case of E. coli, the cell lysis takes place approximately 22 minutes after infection. Most phages synthesize at least two proteins, one of which is a murine hydrolase or lysin, and the other is a membrane protein which is called the holin. The function of the holin is to create a lesion in the cytoplasmic membrane. The murine hydrolase passes through it to gain access to the murine layer and degrades it. The number of phages released per host cell is called the burst size and is usually around 50 to 200 phage particles. Let us quickly revise the lytic cycle of the T4 bacteriophage. It starts its life cycle with attachment to the host cell surface, followed by injection of the viral nucleic acid and degradation of the host DNA. The next step is replication of phage DNA using the host cell machinery and protein synthesis, followed by assembly of new phage particles, and finally lysis of the host cell and release of the progeny.